this is Dr. Dobbs again, and we're going to continue on with Chapter 11, looking at membrane structure, including the lipids that make up the lipid bilayer and proteins that are found within it. Hopefully much of this chapter is a review of things that you've learned before, where we're just kind of expanding or building upon that foundation. When I think of membrane, I sometimes ask students to draw a cell. Right, so if I asked you to draw a cell, my guess is the first thing you draw is a circle, right? And that circle would represent the cell membrane. Most often, the next thing you draw is another circle, the nucleus. And maybe you might draw something like this. My drawing skills on the trackpad aren't great, but as a mitochondria, really bad, but an ER, endoplasmic reticulum. If I ask you to draw a cell, most of you are going to start with a circle and then put a circle inside of it for the nucleus, and you may remember some of the other organelles. When you are drawing a cell with just a line, the line you're drawing is the cell membrane. When you're drawing the nucleus, the nuclear envelope is made of membrane, bilayer. ER is also made of membrane. So membrane or phospholipid bilayer is an important part, or kind of creates the structures of the cell itself. I ask you, what do you draw first? You draw a cell membrane. And again, the phospholipid bilayer is what defines or composes the boundaries of the cell. In a bacterial cell, it's just that simple cell membrane or plasma membrane, whereas we have more internal membrane in a eukaryotic cell. And we'll be working with, or discussing, since we're online, bacterial cells, E. coli, which is a bacterial cell. In the lab, we usually lyse those or break them open, so you're breaking open that plasma membrane. When we think about people, we're thinking of eukaryotic cells with the nucleus, and much more internal membrane that make up the many organelles. The cell membrane has many important functions. So if we're thinking about what happens at the site of the cell membrane, one of the things that happen at the cell membrane is receiving information. The information that's received at the membrane is transmitted through into the cell, and that then can cause changes in behavior. This will be the focus of Chapter 16. The cell membrane is also the site of import and export of small molecules. We will look at that in more detail in Chapter 12. Also, movement of the cell membrane is important in the process of cell movement and expansion and creating cell shape. The membrane expands, creating an expansion of the compartment. We'll say, though, that the actual shape and structure is provided by the cytoskeleton. So this picture is a much nicer one than the little sketch I drew, but you can see more of the organelles within a eukaryotic cell. Right, here's our nucleus with its double membrane. The they're showing very little, but this would be ER too. Mitochondria, transport vesicles. There's a vesicle that's fusing, Golgi, an endosome, so bringing materials into the cell, lysosome breaking down cells, peroxisome kind of containing toxic byproducts from chemical reactions. All of the gray, or this kind of dark gray line throughout the image, is phospholipid bilayer. So it may be that when you think of membrane and phospholipid bilayer, you think of cell membrane, but there's really a large network of membrane within a eukaryotic cell. So we're going to look more at the molecular structure of lipids and how they behave, in particular in water. So phospholipids are the most numerous macromolecules, and they're also the lipid that I think is least familiar to people just kind of from their everyday life. Other lipids you might be familiar with are fats, right, because you think of them in our diet, and cholesterol and steroids are also lipids. Phospholipids, though, are making up or composing the membranes, including our cell membrane and the organelle membranes. Phospholipids are amphipathic, which means they have both a hydrophilic and a hydrophobic pole. Hydrophilic, water-loving, will associate and make hydrogen bonds. Hydrophobic is tucked away. So remember that from the characteristics of amino acid side chains, right? And part of that was whether they had polarity or charge. Things with polarity and charge would be hydrophilic. Anything where there are nonpolar bonds, where there's very equal sharing of electrons, hydrophobic. Here's one molecule that has both hydrophilic and hydrophobic characteristics. You can understand those characteristics maybe a little bit more as we look into the structure of the molecule. The polar end is often called the hydrophilic head. And if we look at the molecules that make up phosphatidylcholine, we have choline, right, in blue. You might notice right here, it's got a charge, right, the nitrogen. And then we have phosphate, which should be familiar from ATP, GTP, right, negative charge. And then we can see bonds in the glycerol between carbons and oxygens, and that would be polar bonds. So within the polar head, we have charge and polar bonds, right, which would make that associate with water. If you look at the chemical structure of these hydrophobic tails, it's hydrocarbons. Carbons and hydrogens which share electrons very evenly. The bonds are very nonpolar. They won't associate with water. In this image, they happen to show you a double bond in one position to show you how that can create a bend in the structure of a phospholipid. So hopefully if I ask you to draw a water molecule, you can do that without too much prompting, right? Water is H2O. So one oxygen, covalently bonded to two hydrogens. With polar bonds, oxygen pulls more strongly on the electrons than the hydrogens. So the polarity of water is very important to how water behaves and other molecules behave with water, right? The fact that there's a kind of a partially negative end and a partially positive end. So how do the molecules behave when they're placed in water according to whether they are water-fearing, water-loving, or have both characteristics in one molecule? So here's an image of a hydrophilic molecule, one that would associate, and we can see that this acetone has partial charges, a partially negative oxygen and a partially positive carbon. And here's that water molecule, just drew with the partial negative oxygen and the partial positive hydrogen. And you can see that we get hydrogen bonds forming between water molecules and also between acetone and the water molecules. The partial negative oxygen from acetone is interacting with the partial positive hydrogen from water. And so these will dissolve or mix in with water. In contrast, this 2-methylpropane is a very nonpolar hydrophobic molecule. It doesn't have any charge or polarity to create hydrogen bonds with water. And so especially if you had more than one of these, they would separate out from the water. And you may be familiar with that if you've seen oil and water, right? You can get that separation of the two. Amphipathic molecules like phospholipids, however, behave very differently and in a very specific way. So each one of these little red ovals with the kind of peach-colored legs, right? Those are phospholipids, and they align in two layers. Right? Here's a single layer here, and here's another layer. They aggregate with their hydrophilic heads facing a watery solution on either side, and the hydrophobic tails kind of in the middle. It might remind you of if you had like a peanut butter sandwich. Imagine the hydrophilic heads being the bread and the peanut butter being the hydrophobic tails. In this image, they're showing you a phospholipid bilayer as a sheet. And the red is the hydrophilic heads and the white is the hydrophobic tails. If you have just a bilayer, you can see there's hydrophobic tails that are exposed. This exposure and this kind of energetically unfavorable condition can be overcome by sealing that sheet into a sphere. So this will happen actually spontaneously if you add phospholipids, a large enough number of them, to water. If we were to go and take the sphere and section or cut through it, we would see this, where we have an aqueous or watery solution in the interior and an aqueous or watery solution in the exterior, with hydrophilic heads facing those aqueous environments and the hydrophobic tails together. And this would be true if this was a cell membrane where we have cytoplasm and extracellular fluid, or if this was within the ER and this was the cytoplasm. No matter what, this would be the orientation or the way that the phospholipids would align with one another. So when you're drawing a cell and you look at an image like this, it seems very static, that the cell membrane looks like a permanent structure and that the shape is just solid, staying in place. But that's not really much like the membrane within a cell, in a eukaryotic cell. The lipid bilayer is actually flexible and acts like a two-dimensional fluid. So we're going to look at what that means. So membranes are fluid. Lipids can move within the bilayer, changing places with one another. And the membranes are flexible, so they can bend. When we think about cellular shape, we may think of the membrane as creating the boundary or the shape that we tend to see or focus on as the boundary, but what actually creates the shape is the underlying cytoskeleton. The membranes themselves are flexible and are kind of like a tent that's being held up by poles. I'm going to run two videos. The first one shows you a technique that can be used to look at the flexibility of membranes, and the second will actually show you how it's used on a cell. So the first is the video of laser tweezers.
The light of a laser beam that is focused into a cone through a microscope objective exerts small forces that can trap particles with high refractive indices near the focal point. This experimental setup is called optical tweezers. It can be used to move small particles, including cells and organelles.
In the second video, we're going to use the laser tweezers to tug on the lipid bilayer of a neuron. You can see a bit of a distortion towards the upper right corner. That's where the computer would be kind of creating a target for the laser. So you would be looking, if you were running this experiment, you would be looking at a computer screen with this slide with the live neuron on it, and you would be able to use the computer to control where the laser tweezers would be working.
To demonstrate the fluidity of the lipid bilayer, a piece of the plasma membrane of this neuronal cell is pulled out with laser tweezers. Remarkably, moving this membrane tubule rapidly back and forth does not rupture the plasma membrane, which flows quickly to adapt to the mechanical distortion.